I was cloning what are called stem cells.
So could you explain what a stem cell is?
I should say that because people don't know. So let's make a simple point. A body is made out of 50 trillion cells. That's a number that is so massive that you can say it, but you can't imagine it. And every day, every one of us is losing hundreds of billions of cells every day, normal attrition. Skin cells are sloughing off. The whole digestive tract, the lining of the digestive tract from the mouth to the anus, this is massive amount of tract. The lining is replaced every 3 days. And I start to say, well, if I keep losing the cells, would I die? And I say, well, if you can't replace them, of course you would die. And I say, yeah, but then I say, how can I keep losing cells and still replace them? The answer is, before you were born, these were embryonic cells that were giving you the structure and function, okay? After you're born, you still have embryonic cells in your body, but you're not an embryo. So we changed the name, call it stem cell, because before birth, I could do a biopsy and look, say, here's an embryonic cell. If I wait 1 minute after birth, look at the same biopsy, look at the same cell, now it's, no, that's called a stem cell. So stem cells are embryonic cells. They're there to replace all the cells that are going wrong. So my research revealed that I was cloning stem cells. And that all of a sudden, what does that mean? I say, I put one cell in a dish, one stem cell. And it divides every 10 hours about. After a week, I've got 30,000 cells in the Petri dish because they keep doubling and doubling and doubling, okay? Here's my experiment, and it was like so clear. I took those 30,000 cells, and now you have to recognize they're genetically identical cells because they only came from one parent. So 30,000 genetically identical cells. I split the cells up into 3 dishes, 10,000 cells in each dish. So all the dishes have genetically identical cells. But we grow them in the lab, something called culture medium. And this, we're going to come back to this. That's critical. Because what is culture medium? I'll give you a hint. Laboratory version of blood. So if I grow human cells, I say, what's human blood made out of? Because that's what nourishes cells. And I make a version in the lab called culture medium. Since I'm synthesizing culture medium, I can change some of the combinations of the chemistry. So I create 3 different versions of culture medium, slight chemistry difference. And I feed the cells. In one dish, I feed them version 1 of the culture medium, in the other version 2, and the third dish, version 3. In the first dish, the cells form muscle. In the second dish, the cells form bone. In the third dish, with a different environment, the cells form fat cells. And here I am teaching in medical school that genes control life, and here's my laboratory experiments to say, all these cells are genetically identical. How come one became muscle, how the other one became bone, how the other one became fat? And it was like, it was based on the culture medium. And all of a sudden I said, oh my God, the cell fate wasn't controlled by the genes, it was controlled by the environment in which the cells live. And if you change the environment, you change the fate of the cells. So that completely canceled the idea that genes were in control.
And that's on the micro. And on the macro, we humans are, as you say, 50 trillion cells or something like that. And our culture medium is the environment around us now.
No, okay. And this is the real important part. Let's look at the body as a skin-covered Petri dish. Why? I got 50 trillion cells under my skin. Instead of a plastic dish, it's a skin dish inside. But I have the original culture medium, which is blood. And I say the chemistry of the culture medium, whether the cell's in a plastic dish or whether it's in the skin dish, it's still the culture medium that controls the fate of the cells. So in the human body, it's the blood chemistry that actually activates the genetics in the cell. And I go, so why is this significant? I say, well, what controls the blood chemistry? It's going to make an important point right here. And I say, the brain is the chemist and puts in the stuff into the growth factors, emotional chemicals, neuropeptides, things like that, adding it, putting it into the blood. And then comes the most important question. So the blood is the chemist putting the chemistry into the blood. The chemistry in the blood is the culture medium. So the chemistry of the culture medium is controlling the fate. Okay, got that. But then the most important question comes, what chemistry should the brain put in to the blood? What chemistry? And then comes the answer, which is so blow away. And the answer is this. Whatever picture you hold in your mind, the brain translates that into complementary chemistry. If I have a picture of love, my brain releases wonderful things into the blood, like dopamine for pleasure, oxytocin to bond with others, a growth hormone. And growth hormone does exactly what it says. So when you're in love, you're releasing this growth hormone, which enhances your vitality. And that's why when people fall in love, they get so healthy. They get, they say, oh, look how they glow. They're so in love. And I go, this is the chemistry of the blood engaging the behavior of the cells. But I say, well, but what if I'm holding a picture of something that scares me? I say, well, you're not going to release love chemistry. You start to release stress hormones and factors that affect the immune system and all that. And I said, well, that's a different chemistry. I go, yeah, just like in the culture dish, a different chemistry leads to a different genetic activity. And all of a sudden I said, oh my God, your thoughts are being turned into complementary chemistry, which then controls your genetics. And this all of a sudden says, well, then if I want to change my genetics, do I have to go in and change the cells in the genetics? I said, no. It's changing your thought. That's all you have to do.
Yeah, and